All right, we're going to go ahead and get started with today's webinar. Good morning and welcome to the Immune Deficiency Foundation's SCID Compass Lunch and Learn. Today, we will hear an overview of breastfeeding and cytomegalovirus from Dr. Jennifer Puck. Before we begin, I would like to point out a few housekeeping items to keep in mind for the webinar. This morning, we are using the Zoom webinar feature. Attendees should be able to see the slides and our presenter and be able to hear us both speak. Attendees will not be able to activate their video camera or their microphone. There will be the opportunity for questions after the presentation. You are welcome to submit any questions you have for Dr. Puck as you think of them throughout the session. Please type them in the Q&A box in the control panel on your screen. Please do not include any personal health information as all questions will be anonymous and read aloud. A brief disclaimer. Please remember, the information presented during this event is not intended to be a substitute for medical advice, diagnosis, or treatment. We are here today as a trusted source and friend to provide you with information. Always seek the advice of your physician or other qualified health provider with questions concerning a medical condition. Never disregard professional medical advice or delay seeking it based on information presented during an educational event. Today's program is offered in partnership with the CALSCID Long-Term Follow-Up Project. And now I am so pleased to introduce our presenter for today. Dr. Jennifer Puck is a professor of pediatrics at the UCSF Division of Allergy and Immunology and Blood and Marrow Transplantation. She treats pediatric patients at the UCSF Benioff Children's Hospital Immunology Clinic and has conducted extensive research with her team there, focusing on newborn screening and gene therapy for babies born with SCID. Dr. Puck also directs the Jeffrey Modell Diagnostic Center for Primary Immune Deficiencies and is the primary investigator for the Primary Immune Deficiency Treatment Consortium. She is also a long-standing member of IDF's Medical Advisory Committee. Thank you for joining us, Dr. Puck.
Well, thank you for having me. And should I share my screen now?
Yep, you can go ahead and pull up your slides.
Okay. Let's see. So can people see them? Can you see them, everybody?
Yep, looking good.
Okay, so I have put my email here in case people have direct questions for me. It's very simple, jenniferpuck@ucsf.edu. And not that I have answers, but I will try to respond. So I thought I would start with just a brief review of the virology of CMV. This is a member of the herpes virus family, along with EBV, the virus that causes mononucleosis, and HSV 1 and 2, and other members. So these are the huge gorillas of the viral world. They are double-stranded linear DNA viruses, and their genomes are the largest of any viruses. And they are very species-specific. So humans are the only source or reservoir of infection. And as you can see in the electron micrograph in the lower left, they have an outer envelope, and that's diagrammed also here on the right side of this slide in the cartoon. And then inside is the nucleocapsid, which contains the DNA. And so these are large and complex viruses. And cytomegalovirus in particular, human cytomegalovirus called HCMV, it's in the beta herpes virus subfamily. And so herpes viruses are the biggest, and CMV is the biggest of the biggest. So its genome is about 325 kilobases, which actually equals 1% of the human genome. So that's enough for dozens and dozens of genes. There are 36 genes related to replication, viral entry, DNA polymerase, and structural proteins. And then many more genes that are encoding proteins for maintaining latency, for becoming reactivated, for pathogenicity, and also evading the host immune system. And many of these genes seem to have been stolen from human cells that were infected along the way by these viruses. So they have sequence homology and resemblance to human genes of the immune system. And so this is a very sneaky, big virus. It has a relatively long reproductive cycle and slow cell-to-cell spread, but that is one way for it to spread in addition to lytic infection. And so it can infect the CMV in particular, can infect many, many different tissue types, not just neurons or B cells or other, more specific, limited tissue types of other herpes viruses. So the transmission of cytomegalovirus is person to person. So infectious virus is found in saliva, urine, blood, cervical and vaginal secretions, semen, tears, and breast milk, just about everywhere. And direct person-to-person contact with infected secretions is the way that people get horizontal infections, such as in daycare settings or between members of a family. Also, blood transfusions can transmit CMV because the donated blood leukocytes, including especially neutrophils, but other leukocytes, harbor CMV intracellularly. And by adulthood, a large proportion of the population has acquired CMV, and different population studies show 35% to over 80% or almost 100% of certain populations, with the higher rates in underdeveloped countries and in areas of poverty. So what are the clinical illnesses that CMV can cause? Asymptomatic acquisition is the way most infant and many childhood infections occur, just with no symptoms at all. But there can be nonspecific cold-like or flu-like symptoms. And so children often become infected with CMV, and it's indistinguishable from the other colds that they get in daycare or school settings. In somewhat older children, teenagers, and young adults, CMV can produce a mononucleosis syndrome that's completely indistinguishable from EBV or Epstein-Barr virus mono. And then significant illness occurs in elderly and immunocompromised persons, including organ transplants and bone marrow transplants. And these include pneumonia, hepatitis, and basically any organ can be infected and damaged. And also there can be a sepsis-like picture. A special category is congenital infection, which happens during pregnancy. And this is very serious when mothers have not had CMV before and they get CMV for the first time while they're pregnant, but also women who've had remote CMV can get reactivation during pregnancy. And so that is another way that congenital CMV can affect the fetus before birth. And it's estimated that about 1 in 200 infants born in the United States are infected with CMV. 1/10 of those have health problems at birth, and half of them will have long-term disabilities. So this is a huge deal. This is the most common congenital infection in the United States, and it's drawn a lot of attention and made people think about newborn screening for CMV and so on. The long-term effects include vision and hearing loss, especially hearing loss, but also seizures, weakness, microcephaly, and intellectual impairment. So it can be devastating. And then we get to early postnatal infection. So now we're going to start to talk about the situation with SCID, which is really close to our hearts. So early postnatal infections include infants with SCID and also infants with other profound T-cell defects. What about CMV in breast milk? So CMV is detectable by viral culture or PCR in colostrum samples and even more so in mature milk samples. And so this graph was published in a paper that followed prospectively a large series of women and did milk samples from very soon after birth to 120 days. And the detection of viral genomes by PCR is called DNA lactea. And you can see that even the earliest colostrum samples on the left can be positive, but the amount of virus there is low. And then the amount of virus goes up by 2 to 3 logs during the next phase, say from 20 to 60 days after the baby is born. And so I think for us, this means that even the very earliest breast milk samples can be positive for CMV, and the amount of CMV goes up over time in general, so that perhaps if we diagnose babies by newborn screening and stop breastfeeding on the early side, that may lessen the risk of breast milk exposure later when there's more CMV. So this is especially dangerous and has been recognized in preterm and low birth weight infants. And very low birth weight is defined as less than 1,500 grams at birth. And these infants have received a lot of attention from neonatologists because when they ingest breast milk that has not been treated, somewhere between 19 to 25% of them develop CMV infection, the majority being symptomatic. But here is a very difficult issue because preterm infants are prone to lots of different conditions, and it's hard to tell what was caused by CMV and what was caused by complications of prematurity. And again, these late effects certainly include bronchopulmonary dysplasia from neonatal pneumonia. But of course, everybody knows that happens in preterm infants also. It's just that statistically, there's considerably more of that, and also retinopathy and hearing impairments in babies who were underweight and who did have CMV. So we still haven't gotten to SCID, and that's because there's really very little in the literature about it. There are certainly case reports of infants with SCID who had CMV who did not do well. And of the studies I've listed here, 2 of the most comprehensive ones. So first is the California experience in their first 6.5 years of newborn screening for SCID. And during this time, there were actually 50 SCID cases identified, but one of them went back home to China and couldn't be followed up, and 49 were followed up. And of them, there were 3 deaths, and 2 of the deaths involved CMV. One with CMV as the only cause, and the other one, an infant who had toxicity from high-dose chemotherapy, but also had CMV. And then the third death in this series was chemotherapy toxicity. So if you look at this, those of us who love newborn screening for SCID have to admit that it isn't perfect. And we need to focus on the cases where, despite newborn screening, we haven't been able to save these babies. And so one of these infants who had CMV was already infected when first hospitalized at 21 days of age. And so I guess the lesson from this is maybe if we could have identified and hospitalized that infant sooner, we could have either prevented the CMV, which may well have come from breastfeeding, or we could have treated it if we had recognized it earlier. The other study I want to mention is from the Primary Immune Deficiency Treatment Consortium, and Jen Heimel is the first author of this multi-author study that included 100 infants with SCID born between 2010 and 2014. So in recent times, but certainly as newborn screening was being rolled out, and certainly wasn't universal. So of these, there were 10 of 59 infants who had a positive history for SCID or had newborn screening. So those were lumped together. And that means 70% of those with screening or history were infected with a herpes virus, either CMV or EBV, or adenovirus was also lumped into this group. However, CMV was by far the most common virus in this group. They didn't break it down further in the paper. And 4 of the 11 SCID patients who died in this study, which is a third of all the deaths, had pre-transplant CMV infection. So I think this shows how serious CMV infection is in SCID infants. And after all, we know that without T cells, without an adaptive immune system, infants have absolutely no defenses against CMV. So how do we treat CMV? Well, ganciclovir is the mainstay, but anybody who's used it knows that it's bone marrow suppressive. And actually, recent data in infants reveals that the standard listed doses are too low to get therapeutic blood levels that can neutralize the virus. And in order to get good blood levels, you really should measure the blood levels, and you end up using a lot more ganciclovir, and that's even more bone marrow suppressive. So you may have to give GCSF or other, GCSF to boost the neutrophil count to try to counteract the marrow suppression. And other drugs include foscarnet, but that does not get into the CNS. Then newer drugs like letermovir and maribavir. So we're very enthusiastic about having new agents with different action from ganciclovir. However, these aren't approved yet in neonates. We use them anyway if we have to, and we're hoping they get approval. And I understand there are studies of these in neonates that are ongoing. And there will be, and there are other new antiviral agents. And also important are now anti-CMV cytotoxic T cells. So these can be given to infants to try to provide some neutralization their CMV while their own immune system is being reconstituted. People have also mentioned things like hyperimmune globulin, and that's not been all that effective. It's not like the case with varicella, for example, where there is varicella hyperimmune globulin that's quite effective. And also the possibility is coming of very specific anti-CMV monoclonal antibodies, but again, little data, and it's not clear whether these could work on their own. So none of these measures really can eradicate CMV infection without the establishment of T-cell immunity. But the flip side of that is that in the face of CMV infection, transplants are fraught with more complications, more graft rejection, and more graft versus host disease. But in any case, urgent transplantation is indicated for babies with SCID when CMV is present. And that means you really have to rush in, even with a haploidentical parental T-depleted transplant with no chemotherapy. This isn't the best way to guarantee establishment of B-cell function later on, but you have to save the infant's life right now. And so this is being done even if people recognize you might need a second cellular therapy later. So now we come to methods that have been suggested to remove CMV from breast milk. And these have been developed with people thinking more of the preterm at-risk infant than the SCID infant, because obviously SCID is such a tiny proportion of the newborn population. So people studied freezing. It appears to decrease the infectivity of contaminated breast milk, but and I guess careful freezing. So the top line here is just the temperature of a regular freezer, and the bottom line is if carefully controlled -20-degree freezer. Sorry, the first line, I guess, is a carefully controlled freezer, and the second line is the regular freezer that might be in your kitchen. And although these reduce the titer of CMV, they do not eliminate it. And so even though this might help a preterm baby whose chances might be lessened of getting CMV by breast milk, this doesn't eliminate it completely. And since SCID patients can't deal with any amount of CMV, this is not an appropriate approach. So people have also looked at pasteurization. The standard so-called Holder pasteurization is 62 degrees centigrade for 30 minutes. And again, this substantially reduces CMV infectivity better than freezing by logs, but it also kills the cells in milk and reduces the nutritional value and inactivates anti-infective factors that are in the milk. So it's also been proposed to use a rapid pasteurization for as little as 5 seconds. And again, that reduces the CMV titer and may preserve some of the beneficial properties of milk. However, it does not totally eliminate infectious virus. And then there are other methods. UV radiation has been proposed, microwaving, exposing to high pressure, and then there are some other proprietary processes with filtration and other kinds of methods that the details of which don't get disclosed. And none of these have been confirmed to totally eliminate CMV by independent testing. None of them are FDA approved. and none of them are widely accepted at the present time. So what do we do? Well, avoiding CMV exposure, of course, would be optimal. So transfusion of infants, including SCID infants, exclusively with blood products that are leuko-reduced, so the white cells are removed, and also irradiated, that does get rid of CMV. So the blood supply can be safe for these infants as long as nurseries follow these guidelines. The methods that I outlined to reduce the CMV in breast milk don't completely eliminate it. And this is my opinion. I think there's no safe enough level of CMV in breast milk for a SCID baby, even if the infectious virus is reduced by log amounts, because again, SCID infants have absolutely no ability to combat CMV, and CMV is such a devastating infection for them. So for infants at high risk, I would say CMV seropositive mothers should not breastfeed infants, and milk from seropositive donors should not be given. And also, newborn screening with TREX should have as short a turnaround time as possible so that we can catch babies before they become infected. And in California, and I think many states, there is a panic positive or urgent positive when the TREX are undetectable or very, very low. And what we do with these babies is ask their mothers not to give them breast milk pending our immune evaluation. So even when the TREX come back, we ask in this situation for the breastfeeding to be held. An anti-CMV vaccine would be great, and it's been worked on for over 40 years, and we don't have one yet. But then again, vaccinology has taken a leap with COVID, so maybe we'll get one soon. And I'm going to stop there. These are the references that I used. I started that Osterholm review because it's quite comprehensive, and I'm happy to take questions, and also happy to be informed by other people who might know more about this than I do.
All right, thank you so much, Dr. Puck. And we know that you have to get going to clinic, so we will be quick with our questions. We do have one in the queue so far. This individual wants to know, does congenital CMV infection present differently from breast milk or other acquired CMV in newly diagnosed SCID patients?
And this is a great question, and the answer is yes. Congenital or in utero CMV infection is already symptomatic at birth and even before birth. And these babies have, they can experience retarded brain development, microcephaly, low birth weight, and other effects of CMV even before they are born. So that is a very serious and devastating sort of situation. And that has prompted a lot of people to say we should develop a vaccine and give it to pregnant women. And that would be wonderful. So this is in its most severe form. Actually, many pregnancies are lost with congenital CMV, and even in less severe forms, babies are born already infected and can have deafness that isn't even apparent right at birth, but becomes evident later, so that CMV is actually now recognized as one of the primary or leading causes of deafness or hearing loss in infants. So after birth, these things can occur, but The developmental effects like the brain development and microcephaly are perhaps less severe, although seizures and postnatal neurological complications certainly occur.
Thank you, Dr. Puck. We're at 11:31. Do you have time for one more question, or do you need to go to clinic?
Sure.
Okay, awesome. Thank you for hanging around for us. All right, this individual wants to know, should a woman who acquired CMV as an adult refrain from breastfeeding her child to avoid passing on a CMV infection?
Well, so breast milk is the best food for babies, and everybody recognizes the benefits of breast milk. And so it's not, it's almost against motherhood and apple pie to say you should not breastfeed. And I agree that breast milk is the best nutrition, even though most people have CMV. Babies who are born at term or who are not, low birth weight or sick or have SCID, they handle CMV, and most of them are, if they are infected, are asymptomatically infected and never experience any complications. So I think that adults who have a history of CMV don't really have to worry if it's a remote history, as long as their baby is not preterm. If adults have a primary infection when they are pregnant, then that's something that their obstetrician needs to jump on and treat with antiviral medication. So it really, it depends on the situation.
Thank you, Dr. Puck. Do we have time for any more, or do you need to go?
There's time for one more.
Okay, thank you so much. All right, last question, everybody. This individual would like to know, are SCID babies more at risk for congenital CMV than unaffected babies?
Well, so having SCID and having congenital CMV is kind of like getting two lightning strikes in the same tree. Both of those things are very rare. So for babies with SCID who have CMV, it's almost always postnatally acquired. And we actually assume, unless we can prove otherwise, that the infection is acquired after birth. And that's why we are suspicious of breast milk as a route of infection. It's just that congenital CMV is so rare, then to have SCID on top of it, while it's not impossible, is extremely unlikely.
Sure thing. Well, thank you so much, Dr. Puck. We know you need to get going to clinic, so thank you so much for taking the time to join us this morning and share your expertise on this topic. We'll let you go and hope you have a great rest of your day.
Okay, thank you.
All right, thanks, Dr. Puck. All right, and to our audience, we have just a couple more items to get through before we close out. We hope that you'll remember to take advantage of all of the additional resources that the SCID Compass program and IDF have to offer. If your question wasn't addressed during the program, go online and submit your question to Ask IDF. Visit www.primary.immune.org/ask-IDF, or you can give us a call at this 800 number. And let us know your question, and our community resource navigator will answer it for you. We also want you to check out the IDF Resource Center and YouTube page to find videos, resources, and more. As we head into fall, IDF has programming for everyone, no matter your connection to PI. Our next IDF webinar will take place on Thursday, September 21st, and will feature an update from Dr. Jude Hajar on her research into PI and the microbiome. And we hope you'll join us again next month for an ADA SCID gene therapy update with Dr. Cohn from UCLA. Be sure to follow us on social media for the latest news and updates. We also have 2 in-person PI Foundations events coming up. one in Chicago on September 30th, and one in Anaheim, California on October 14th. And finally, we hope you will save the date for our 2024 PI conference, which we are so thrilled to share will be hosted in person at the Sheridan Grand River Walk in Chicago from June 20th to 22nd of next year. We want to again thank the CALSCID team for partnering with us on today's program and give a big thank you to Dr. Puck for sharing her immense knowledge on this important topic. Thanks for joining us and have a wonderful rest of your day.